Thank you. My name is Philip, and I'm presenting on behalf of Dr. Allison Petrini. It is my pleasure to share with you our study on ooplast-mediated male genome replication and how we use these cloned male gametes to generate developmentally competent zygotes. First, I'll provide you some background information. Male genome can be propagated using cellular machineries of an oocyte through a process known as androgenesis, when the sperm genome becomes the sole source of the nuclear genetic material in the developing pseudoembryo. Resulting blastomeres with cloned haploid male genome, also known as androgenotes, have the same genetic components of the original spermatozoa, and they can be utilized as the source of male pronucleus to complement the oocyte. Moving on, there are two major aims for this study. The first aim is to replicate the male genome of a single spermatozoon. The second aim is to utilize the cloned male genome to complement a recipient female genome and to generate developmentally competent zygotes capable for full pre-implantation embryogenesis. In order to accomplish the aims, here are the key steps of the study. First, the design of the study involves the generation of a haploid male embryo. We allow it to cleave in order to replicate the sperm genome. We then synchronize the cell cycle of the haploid androgenotes by temporarily inhibit its DNA polymerase to arrest them at pre-S phase before DNA synthesis. Next, cell cycle synchronized androgenotes were isolated and fused with a recipient oocyte to construct a biparental zygote. And lastly, we assess the embryo development of the reconstructed zygotes and compare to control conceptuses generated by standard piezo-actuated ICSI. Here, let me introduce a timeline on how we replicate male genome and how we prepare for biparental zygote reconstruction. From the beginning at hour 0, a cohort of mouse metaphase 2 oocytes were retrieved. The oocytes are visualized under polarized microscopy to identify the spindle complex. It is then enucleated. The resulting ooplast is injected with a single mouse sperm head. 6 hours after incubation, a male haploid pronucleus will form. 4 hours later, at hour 10, another cohort of mouse oocytes will be retrieved as the source of the female genome. These intact oocytes will be activated chemically. 6 hours after the activation, at hour 16, the oocyte extruded a second polar body and with a single female pronucleus will be selected as a recipient. Meanwhile, also at hour 16, the cleavage of the male haploid embryo will be confirmed. Those cleaved embryo carrying duplicated copies of the male genome will be selected for reconstruction. Then a single male haploid androgenotes will be isolated and transferred into the perivitelline space of the recipient oocyte. Followed by fusion, the reconstructed biparental zygote will be monitored and cultured. To continue, I will provide some details on how we obtain male haploid embryos. First, a cohort of oocytes were retrieved from superovulated B6D2 generation 1 adult mouse. These oocytes are exposed to cytokeratin B in order to re-elect the cytoskeleton and enhance the flexibility of the oocyte. Those oocytes are viewed under oocyte polarized microscopy to identify the spindle complex and enucleated with micromanipulation. Followed by that, piezo-actuated ICSI will be performed to inseminate the resulting ooplast. And this is how we conduct each steps for the male haploid embryo. This is how an oocyte looks under a polarized microscopy. The red arrow indicates the spindle complex. The spindle complex is then suctioned by the micropipette. And here are the resulting ooplasts inside their respective zona pellucida. As you can see, the caroplasts containing the bright spindle complex are adjacent to the ooplast. We then perform the piezo-actuated ICSI by injecting a single sperm head into the ooplast. The resulting male haploid embryo will demonstrate a single pronuclei, as shown in the blue circle. And 16 hours after the insemination, this is a cleaved male haploid embryo. Each cleaved blastomere contains an exact cloned copy of the sperm genome. To continue, now I'm going to describe how we prepare for the recipient oocyte. Another cohort of oocyte will be retrieved from the mouse strain that is exactly the same as the one we retrieved for the male genome cloning. The oocyte will be exposed to the calcium ionophore to induce calcium oscillation of the oocyte. The successfully activated oocyte will extrude a second polar body and resume its cell cycle and form a female pronucleus. And here is how a paternogenically activated oocyte looks like. Notice the red circle, which encircles the female pronucleus and two polar bodies on 12 o'clock. To prepare for zygote reconstruction, the male haploid embryo were exposed to acetylcholine immediately after the first zygotic cleavage to temporarily inhibit the DNA polymerase. Next, a single male androgenotes were isolated from the male haploid embryo. And to construct a biparental zygote, a single androgenotes were transferred into the perivitellin space of the recipient oocyte. The fusion of the recipient oocyte and the male haploid androgenotes are mediated by Sendai virus. Here is how we perform a biparental zygote reconstruction. A single male androgenotes inside the pipette is transferred into the perivitellin space of the paternogenically activated oocyte. Notice the pronucleus and the two polar bodies. And here is a reconstructed biparental zygote with two pronuclei in the same cytoplasm. Next, the zygote will be cultured in a Timelapse incubator up to 96 hours for full pre-implantation development. The cleavage parameter and the development characteristic will be compared to the control conceptusis generated by standard piezo ICSI on unmanipulated oocytes. Here, I will present the entire development of both control and the reconstructed zygotes side by side. Starting from hour 5 after the manipulation, the syngamy occurred at hour 14, followed by first cleavage. Further timing of the cleavage into 4 cells and the eventual 8 cells are also comparable. Next, the developmental into compacting morula and the blastocyst are also similar. Both control and experimental zygotes are capable to undergo hatching. Moving on to the result of overall embryo development. In this preliminary study, the development of the reconstructed zygotes into 2-cell, 4-cell, morula, and the blastocyst are comparable to the control zygotes. And here is a graphic representation comparing cleavage between control and the reconstructed zygotes. The control reached an eventual 80% blastulation rate. The reconstructed zygotes resulted a 76.9% blastulation rate, a slightly lower but comparable to the control. To conclude, this study has shown that the ooplast-mediated male genome cloning is feasible in a mouse model. Those replicated male genome can be utilized to construct a developmentally competent zygote able to achieve complete pre-implantation embryogenesis. Once this technique is optimized in human, it offers the possibility to replicate precious male gametes, especially for men with severe hypospermatogenesis. Moving on, this technique may also allow pre-fertilization gamete screening on a single spermatosome. For example, one androgenous can be used for reproductive purpose, while the sibling spared one can be used for genetic screening. It can help couples presenting with gamete heterozygosity. Indeed, this technique can be utilized in conjunction with experimental heritable genome editing. This will be possible by cytoplasmic-mediated sperm genome decondensation and eventual replication.